So this is our Punnett squares, lesson 3. We are talking about multiple alleles and sex-linked inheritance in today's lesson. With multiple alleles, when you're thinking of your gene controlling your trait, multiple alleles means that there's more than two options. There's many options. In our last lessons with our Mendelian Punnett squares, we were saying like, okay, it's either red or blue. With incomplete dominance, we had where it was still just with two alleles, but it had a variation in the heterozygous. So there's 3 phenotypes for those ones, controlled by 2 alleles. But this one, there's many alleles, and therefore there's many combinations for the same gene. So it's all gene C, but the gene for C could have, it could say just C. It could say C with a superscript CH or C with a superscript H, or it could have as a recessive lowercase c. So with the fur of the rabbits here, big C would be the genotype, and the phenotype would be for brown fur, which is called the wild type. Then you have C with the superscript CH. A combination of those two would be a chinchilla. And then if you have the C little, or the C with little h, it would be the Himalayan. And then the recessive is albino. So the only time you could be recessive is if it's only lowercase c's. And there's differences with those dominant and recessive. So for that situation, the brown coat is dominant. And it's dominant over chinchilla, and it's dominant over Himalayan, and it's dominant over albino. So as long as you have a big C, it's going to always show the wild type. And then C with the superscript CH is dominant over the Himalayan, it's dominant over the albino. So it could say CCH and then CH or CCH and little c, and it'd still show chinchilla and so forth. So the Himalayan is dominant over the albino, but it's not dominant over wild type or chinchilla. You can find multiple allele characteristics in many different organisms, and we define it as the alleles are the same, then the organism is homozygous for that trait, if they are both alleles for brown eye color, for example. And then when alleles are different, then the organism is heterozygous for that trait, and the dominant trait is expressed. One allele is dominant brown eye over the recessive, which is blue. With multiple gene inheritance, this is like hair color, skin color, height, among many other traits. They're controlled by multiple genes. Therefore, multiple alleles control them, and they work in combination. So this explains why there are many variations among the individuals. One common example we have is our blood typing. For our blood type, we're talking about the surface proteins on our red blood cells. The particular gene of interest is the ABO gene. We also sometimes talk about the RH gene, which makes you positive or negative. But for now, we just look at this one gene, ABO. If both parents pass you an A, it would make you type A blood. On the left side of this diagram, you can see this combination. If one parent passed you an A and the other one passed you an O, then you're going to see that you're still type A blood. These ones are type A here. And then if one parent passed you an A, but the other parent passed you a B, you are type AB blood. This is a co-dominant situation. So A is not dominant over B, and B is not dominant over A. They can express themselves at the same time. If both parents passed you a B, so being homozygous for type B, you will be B blood. If you have one parent passing you a B and the other parent passing you an O, you're heterozygous for your genes, you would still be B blood because B is dominant over O, just like A is dominant over O. So O is our recessive one, and the only time you can be O is if your genes only say O. So there's many variations of the gene. We looked at scenarios where there's one dominant allele, one recessive allele, or there's like 3 phenotypes. Today we'll be looking at where there's 4 or more. And the variation within the species, it helps to ensure that the survival of that species. So we don't want everything to be cloned exactly the same. We do want variation, but this helps with diversity in your population. And if your DNA is too similar, it ends up with problems later on. But so multiple alleles is when there are more than 2 alleles for a specific trait. Remember that parents only have 2 alleles they can pass on to their offspring. However, when 4 or more phenotypes exist in a population, then there must be several different alleles. There's more than 2 at least. And that is our classic example of blood typing. Color in cats, fur color, and their height is an example of, and their length is multiple alleles. They can be, the basic fur color is black, brown, orange, red, white, but they could have no hair, they could have short hair, they could have long hair, they could have medium length hair, they could have wavy hair, straight hair, curly hair. And this is another way you can write the blood types on eyes. This is the same gene, it's just the variations of that gene. So we could have IA, which is homozygous A. IA little I is heterozygous for A. IB is homozygous for B. IB little I is the same thing as saying like BO, and that's heterozygous for B. IAIB is the same thing as saying AB, and that's your codominant AB scenario. And then little I is the same thing as saying O, and that is your O scenario. Think about what other domesticated animals have many different fur colors, textures, lengths. Think of the variety that you find in some organisms. You probably can think of a lot of examples for that, but there's little video segments you can watch from the Nuba sisters, and then there's questions that you'll be able to answer. And that one has 2 choices for your answer. And this one's asking you about the genotypes for type AB blood. There's only 1 choice for that one. This is asking you the genotypes for type A blood. There's 2 choices for this one. Asking you the genotypes for type O. There's only 1 choice for that one. Our blood, every person's blood does the same thing. It's going to be a transporter, it's going to be a regulator, and it's going to be a defender for your body, transporting nutrients, getting rid of waste products, whether it's to your lungs, your kidney, your spleen. It's going to help to maintain homeostasis for your body, and it's going to help to defend your body against infection. Your blood is a complex tissue made of many cells. Red blood cells, one of them. You also have white blood cells, platelets, and then the fluid portion of your blood is called plasma. These are showing you the red blood cells, and these are showing you the surface proteins called antigens that give you these unique characteristics. Just like you have unique characteristics to your body, your hair color, your eye color, if you have freckles or not, your red blood cells have unique characteristics to their red blood cells too, like so it's sticking out of their cell. So for type AB blood, you would have both A and B antigens. Type A blood would have just the A antigens. Type B blood would have B antigens, and the type O blood has neither A or B antigens. That's why type O blood is technically a universal donor, because they can give to everyone else. They're not going to cause an immune response. The other ones are universal donors because they do cause a response. There's another type of protein in your blood called antibodies. They float around in the plasma to help protect you, and they look like little pitchforks. Type AB blood actually has no antibodies, but type A blood has antibodies, they have a B antibody, meaning they don't want B blood. They're going to block the B blood. Type B has A antibodies. They're going to block A blood. They don't want to accept A blood. And type O blood has both A and B antibodies. They do not want either A or B blood. What's kind of cool is type AB blood, they can accept everybody because they don't have antibodies. And therefore we call them a universal recipient. Now there is another protein involved that we usually talk about with compatibility. There are actually many different surface proteins on your red blood cells, over hundreds of them, but these are the two most common ones that we talk about in biology because they cause a lot of problems with compatibility compared to the other ones. The Rh factor, first discovered in rhesus monkeys, and then they found it in all other mammals, but it's either like if you have it, then it's going to make you positive. If you don't have it, then you're negative. If you don't have it and you're negative, you actually have antibodies to block it. So the negative blood cells would have antibodies against the positive Rh, meaning that their body is going to protect them against that. They cannot accept people who have positive blood. The negative people can donate their blood to positive people as long as their surface proteins match, but positive people cannot donate to negative people. When you're donating blood, you can accept blood. If you're positive, you can accept blood from positive and negative people, and you're going to accept people's blood who has at least what you have on your blood. For example, AB positive people have A antigens and B antigens and the Rh antigens. They can accept people who have just the A antigens or just the B antigens or no antigens or just the Rh antigens. They can accept the A and the Rh, the B and the Rh, or just AB antigens. They can accept themselves as well. That's why they're a universal acceptor. They can accept everybody because they have all these surface proteins. In comparison, a negative blood can only accept A negative blood and O negative blood. They don't want anything with the Rh. Their body can't accept that. They also do not like the B antigens. You can see those there. How does this relate to our Punnett squares? Well, you're able to kind of analyze what the possible combinations of the child's blood would be if you know the parent's blood. In this problem, it says, let's cross two individuals. One is heterozygous for the A blood type. They put the parent up top here saying IA, little I. You could also write that as just AO, and it gives you the same results. And the other parent is heterozygous for type B, IB little I. And therefore, we have BO. So you'd carry the A down and the O down, the B across. And we want to put these chronologically with the alphabet just so they're easier to calculate. You bring the IA down, and you want to keep the superscripts with the capital I's. They travel as a whole unit there. The IB is going all the way across. The little I is going across. And whatever square you tend to use, you'll come to the same conclusions. For your phenotype ratio, we would say how many of each type we have from the square. So what's the chances of the child being type A, the child being type B, the child being type AB, the child being O? For the genotype, you would list what's from your squares. Some people like to write them all out. So they would have like AA, AO, AB, BB, BO, OO. Put this one as an AB. We have a BO, an AO, and OO. We get the same thing for that square. our calculations. Anything that says AA or AO would tally up for the phenotype type A. Anything that says BB or BO would tally up for the phenotype for B. Anything that says AB would be the AB phenotype. Anything that says OO would be the O genotype, which would be the same thing as little I. AB is the same thing as saying like IA, IB. BB is the same thing as saying IB, or IB little I. AA is the same thing as saying IA, or IA little I. So you'd have one type A, one type B, one type AB, and one O. Or a 25% chance of the child being any of the 4 blood types. And each time they have a child, the squares would reset. Therefore, just because they had a child who's type A blood doesn't make it more or less likely for the next child to be A blood as well. So the squares just kind of reestablish themselves. A couple multiple choice questions. This chart's nice because it shows you like who can donate and who can receive blood. Showing you that visual. So donations going down. O negative, they can donate to everybody. But O positive can only donate to the positive blood types. And then when you're receiving blood, it's going across. Like AB negative can only accept blood from the negative blood types. They can accept from O negative, A negative, B negative, and AB negative. They do not want the positive RH. We have a couple more multiple choice questions. There's 3 choices on that question. There's a little video clip you'll be able to watch in a couple questions. Go ahead and take your time and answer these questions. You have several here. And then we will talk about succulent genes. We have 2 types of squares today. Sex-linked traits are found on the sex chromosomes. You have 46 chromosomes total. The first 44 are called autosomal chromosomes or your body cell chromosomes. And the last pair, the last 2, 45 and 46, those are your sex cell chromosomes. They're going to help determining characteristics for the egg and sperm cells and for those hormones involved that are regulated by your organs and everything. The female sex chromosomes we traditionally write as XX and the male's XY. There are other combinations, but these are the most prevalent ones. So inheritance of characters by a single gene may deviate from simple Mendelian patterns. When alleles are not completely dominant or recessive, when a gene has more than 2 alleles, when multiple genes influence a single phenotype, or when the environment impacts the phenotype, or when genes are not located on nuclear chromosomes. Thomas Hunt Morgan discovered that genes are located on chromosomes at specific locations through his study of inheritance in fruit flies, the dystrophia. His findings provided molecular mechanisms for the model of inheritance that Mendel came up with. Chromosomes explain how genes are made up of 2 alleles and how those alleles separate and insort themselves independently of other genes during the gamete formation, during meiosis. The sex of an individual, of an offspring, is actually determined by the male sex cell, the sperm cell, because the male could pass an X on to produce XX for if having a daughter, or XY, which would traditionally produce a son. Now, again, remember, these are just the common scenarios. There are other combinations that could come about. Like, for example, there's a situation where if non-disjunction takes place, or people could be XXX or XXY or XYY. We're going to look how to set this up real quick, and then we'll continue on. All right, you are viewing the whiteboard now. We usually are tracking just complete dominance with our sexing traits. A common example is colorblindness. And colorblindness is sex-linked recessive. For you to be colorblind, you have to have the recessive genes on your sex cells. For a female to be colorblind, we would have X. We're going to use A's because I can't make big and little C's. We'd have to have X little a for the female to be colorblind. For the male to be colorblind, he only needs one little a because most of the sex-linked traits are carried on the X chromosomes. There are Y-linked ones too, but not as common. And that is recessive. For you to be normal color vision, for a female, there's actually two options. You could be X big A, X big A, or X big A, X little a. And sometimes you hear this talked about as being a carrier, X big A, X little a, because she has normal color vision, but she could actually pass X little a on to her offspring, potentially making them colorblind, even though she's not. And for a male, the only way that you can have normal color vision is if you have X big A, Y. What you would do is you figure out what the parents' genes are and you'd put them on the square. We're always going to have one parent up top and one parent on the side. Let's say we cross the dad who's colorblind with a mom who is a carrier or heterozygous. You would carry the letters across and the A's stay with the X's. They travel as a whole unit. My X over, to put it chronologically. Oh wait, Y. And that says X little Y. After you do your squares, then you're going to tally up how many are normal versus colorblind for the phenotype, and you can list out all their genotypes. These two will be showing colored vision. These two would be colorblind. There's a 50-50 chance there. Sometimes they'll ask you like of just the males. If they do ask you of just the males, how many are colorblind, that means you're only going to look at the boxes that are X, Y. And it's still 50-50 chance, but it is taken out of 2 out of the X, Y boxes instead of 4. So sometimes they'll ask you to analyze. So this one's showing you this square, and it says, what's the percentage of female offspring that will be a carrier for the recessive disease? So they want to know how many are carriers or heterozygous. So how many are X big A, X little a? This just shows you that the egg can donate only an X, but the sperm can donate X or Y. Therefore, the sperm returns the sex of the child. This just shows you the fruit flies, red-eyed versus white-eyed. And he was able to determine that there are sex-linked characteristics based on the fruit flies. Pretty much he was looking at the eye color as monohybrid crosses, single trait crosses, and he was able to see that it is linked. It was more common for males to have white eyes as a sex-linked recessive trait than females because they ended up only needing one gene in the end, one allele to provide that white gene, but females needed both alleles to be that. What type of antigens can be found on the surface of red blood cells when someone has type O blood? I'm not sure why my questions are scattered here. When he crossed pure red, white-eyed males and red-eyed females, he was unable to produce a female with white eyes. And he concluded that the genes must be located on the X. So some of those traits are located on the sex chromosomes, and the parents determine those traits being passed on. This is a question. Most known sex-linked traits are X-linked. They're carried on the X chromosome. And that's probably because there's just a lot more genes on the X chromosome. The Y one is quite small compared to the X. Some of them are associated with disorders. Not all of them are, but males are much more likely to be inheriting the sex disorders because they only inherit one X. So if the X has the allele for the disorder, they will suffer from the disorder. It's sometimes talked about as recessive lethal X-linked traits, and it results in death. So you'll have some questions again. And this is showing you the Punnett square setup, crossing a homozygous dominant female with a recessive male. And you just assume they're X-linked unless I tell you they're Y-linked, but most of the time they're X-linked. And hemophilia is a recessive X-linked trait. They want to know what's the probability of having a hemophilic child if the man does not have hemophilia, but the woman is a carrier. So the man does not have hemophilia. We'll just have him as X big HY. And the female is a carrier, so she'll be X. Here's a bug. A little HY. Sorry about that. A bug just hit me in the face. Okay. So back to this. So the male does not have hemophilia, but the female is a carrier. Then we would bring our X big H down and the Y again, the X big H across, and the X little h across. And then you'll be able to decide how many of them have hemophilia or how many have no hemophilia. So hemophilia is recessive. In order for them to have hemophilia, the female must say X little h, and the male must say X little h, Y. No hemophilia is dominant. For a male to have no hemophilia, he will say X big HY. And for the female to not have hemophilia, she would say X big H, or X big H, X little h. So you'd calculate how many of them fit those categories based on the square. There's also Punnett squares continue on to pedigrees. So if you're looking and to exploring pedigrees, they're kind of cool. They kind of just give you another visual representation, but it's not just parent and offspring, it's the whole family. It's like a big family tree. You can see grandparents, you can see siblings, aunts and uncles. The circles on a pedigree represent a female, and the squares on the pedigree represent a male. Anything shaded in is going to be the ones affected. If you see things that are half shaded, it means that they're a carrier for that trait. When you see that it says autosomal dominant, autosomal means that it's not sex-linked chromosomes. So it's going to be like one of the first 44 or 22 pairs of chromosomes. And you don't have to use XX or XY. And only when it says it's sex-linked do you have to use XX and XY. When it's autosomal dominant, every affected person should have at least one affected parent. Males and females should be equally often affected. And an infected person has at least a 50% chance of transmitting the dominant allele to other offspring. For example, in our first generation, the top branch here, so if there's Roman numerals off to the side, those are the generations. The lines that connect the shapes means they're married, and the branches that come down from the married line are their offspring. The shaded in square on the top row here in generation 1, this male, this is the dad here. His genes would be like either big B or big B little b. Based on his children here, we're able to decide that he's big B little b because he's married and his wife is little b. This is a dominant situation. So we know she doesn't have the trait, so she's all little b's. We know that she's passing little b's to her children, and we know that he's passing at least a big B to the ones that are shaded in. And we determined that he's big B little b because he has a daughter who is not affected by the trait. So you kind of just work your way around. Now, sometimes you won't actually be able to decide what their genes are. For example, if you don't have any one above or below you to decide, like if they don't have any children or anything, we might not know their other gene. We know they have a big B, but we don't know if they have big B, or big B, little b. Autosomal recessive refers to the situations where two recessive alleles result in the trait being expressed. You sometimes will see, they might use 2 different colors instead of half shades, but those would technically be half shaded, the purple ones. Those are carriers. They'd be like big A, little a. And they don't actually show the recessive trait themselves, but they do pass it on. At least they could pass it on. And in autosomal recessive inheritance, you do see it's much rarer in the population. It tends to skip generations. An infected person may not have affected parents, and parents would be carriers. Because even though they are not showing the trait, their genes still have at least one little letter. That's why they don't express it, because they have the dominant letter too. It affects everyone equally, and it tends to skip generations. Two affected parents will have affected children 100% of the time. So if both parents are shaded in, all their children will also be shaded in. Here's an example of autosomal recessive. Here's a codominance impairments example. Sex-linked recessive, it's much more common to be a male who's affected than a female because they only need one allele to have it. So X little b Y, for example. Males have an 8-to-1 ratio of being more likely to have it than females. And they transmit the alleles to all their daughters, but none of their sons, because a male would pass the Y to the sons. Homozygous recessive females can arise only from mating in which the father is affected and the mother is affected, or she's a carrier. So she'd have to be like X little H Y, X little H, X little H, or X little big H, X little H, cross with X little H. And sexual dominant. Is there a single dominant allele on the X chromosome can lead to trait condition? There's a lot of them there. Twice as many females are affected than males in this situation, and it's usually half the children of an affected female will be affected regardless of the sex. Vitamin D resistant. Remember, these are chart-shown relationships between members of the family. Squares are the males, circles are the females. The colored in shapes are the people affected. The half-shaded shapes, or the lightly shaded ones in some of the situations, are showing carriers. You'll be able to answer the rest of these questions on the quizzes for practice, and then you do have other references that you can look into. There's vocabulary Quizlets and worksheets you can do. Okay. And then there's a Nina Sisters video on worksheet if you like that. But that's all I have for you today. Let me know if you have any questions or anything, and I'm happy to help. Thanks for joining me today. Bye, everybody.